Welcome, everyone. Thank you for tuning in to this month's Fireside Chat in honor of Colorectal Cancer Awareness Month. The Fireside Chats are brought to you by the Office of Community Outreach and Engagement at the Masonic Cancer Center at the University of Minnesota. The Office of Community Outreach and Engagement serves as a bridge between the Cancer Center and communities across the state of Minnesota. Our team's mission is to reduce the burden of cancer in Minnesota by engaging communities and providing them access to knowledge and information on the latest cancer research, cancer prevention, the importance of screening and early detection, treatment options, survivorship, as well as clinical research opportunities. My name is Amna Hussein. I am the Community Engagement Coordinator on our team, and on behalf of our team, thank you all so much for joining us today to help raise awareness about colorectal cancer and the importance of screening. Colorectal cancer remains the most preventable yet least prevented cancer in our country. It is thought that about 98% of colorectal cancers and deaths are thought to be preventable. So today we are joined by two incredible speakers. We are joined by Dr. Subree Subramanian and colon cancer survivor Mike Neeson for an important discussion on colon cancer, the latest on colon cancer research at the University of Minnesota, and hear the colon cancer journey of a survivor, Mike Neeson. First, we will be hearing from Dr. Subree Subramanian. Dr. Subramanian is a professor in the Department of Surgery at the University of Minnesota. He leads an internationally recognized cancer research program that focuses on the molecular mechanisms of anti-tumor immune evasion in cancer, which he will actually be talking about today. His current research explores the interactions between cancer and the host immune system and how cancer cells manipulate the anti-tumor immune response. With over 120 peer-reviewed publications and numerous book chapters and review articles, Dr. Subramanian has made significant contributions to the field of colorectal cancer research. He has also delivered over 80 invited lectures, seminars internationally, and was awarded the American Cancer Society Research Scholar Award. I will introduce Mike later on in our program today. So we will be recording today's session as we do with all of the fireside chats, and you can refer back to them on our YouTube page. That's at Masonic Cancer Center. Before we begin, I would like our audience to get familiar with using the Q&A feature, which is going to be located on the bottom right of your Zoom screen. During today's session, please use the Q&A feature to submit your comments, questions, reactions, and we have time carved out at the end for some Q&A. Okay, now I will not continue anymore, and I'll pass it over to Dr. Subramanian. Dr. Subramanian, the floor is all yours.
Thanks, Amana. I think this is wonderful to meet everyone virtually, and especially this month being a colon cancer awareness month. This is a wonderful time to really connect with the community and then to talk about our research so that everyone knows in terms of what's going on in terms of research activities, specifically on colon cancer at the University of Minnesota Cancer Center, as well as in the Department of Surgery. So with that, I'm going to share my screen to see if we can you see my slides here?
Yes, just not in presenter mode.
Oh, wait a second. Okay, so
Perfect.
Can you see my slides?
Yes.
So what I'm planning to do is in the next maybe like 15, 20 minutes to kind of briefly walk you through the various research aspects that we work in the lab, primarily on colorectal cancer. So I'm Dr. Subree Subramanian. I'm primarily one of the faculty members in the Department of Surgery and also involved extensively in the Masonic Cancer Center. So as many of you know, colon cancer is the second leading cause of cancer-related death in both men and women. Like Amina pointed out, this is pretty much preventable, but still we are not doing everything that within our limits to kind of do screenings and things like that. But we are going to kind of take and discuss those aspects. But this year alone, there will be 150,000 new cases being diagnosed with colon cancer, and of which 54,000 deaths will be happening in US alone. That's a mind-boggling number, so which is pretty much preventable. Worldwide, 1.8 million new cases will be diagnosed this year, and maybe every year. So that's also again, which is something which we have to really think about it and worry about it. More worrying feature is increasing trend of early onset of colorectal cancer. Individuals who are below 40 and even at the like mid 30s, they are being diagnosed with like high-grade colorectal cancer, and many times their outcomes are not favorable. So we are trying to understand what are the mechanisms and then what could be contributing to the early onset of colorectal cancer. This is something which everyone is really thinking in colon cancer, in both in clinics as well as in the research side. So in the last 60 years or so, since we started like specifically treating with chemotherapy combinations, 5-fluorouracil has been one of the standard of care. And as many of you know, this drug, specifically 5-fluorouracil, was introduced in 1962. It's almost 60 years. We are still using the same 5-fluorouracil and its combination with other chemotherapies. So for the last 60 years, a lot of development has happened, but still there's a long way to go, really understanding various mechanisms of cancer development and how it evades immune response and all those aspects of tumor development and progression. And the last 10 years or so, there will be targeted therapies, specifically focusing on EGFR inhibitors and also targeting the angiogenesis with the VEGF receptor targets. So this is again like targeted therapy. There are not many targeted therapy available at this date for colorectal cancer. Specifically, these are the two major things at this time in the clinics. With all these things said, this is something which in the last 5 years we are thinking about immunotherapy, which has really changed the way we treat cancer patients in general, especially when it comes to colorectal cancer. Patients with microsatellite instability, who are like kind of deficient mismatch repair, meaning they have a mutation which happens spontaneously, those mutations are not repaired. Those patients are really a candidate for immunotherapy. And if you look at the last 4 months, or like this in terms of publication, what happens in this domain, a recent clinical trial using this dostarlimab, which was given to rectal cancer patients who are in a kind of advanced stage disease. These are like early stages of clinical trial where there are 14 patients were treated with the dostarlimab. And in the follow-up, up to like 12 months, none of these patients really kind of developed any cancer. There's no recurrence or no disease progression. So all the 14 patients who are treated with this dostarlimab, which is an immunotherapy which targets PD-1, a gene which prevents T cell functioning, all these patients got cured of their disease. And there was a follow-up clinical trial based on this. And there included more patients, and everyone showed a complete response. This was never heard of in the cancer treatment world, that all the patients in a clinical trial got complete response. This really kind of gave us a lot of hope that immunotherapy is something that most patients can benefit from. At this point, dostarlimab is given again only to the microsatellite unstable patients who have rectal cancer. So this is pretty much for mismatch repair deficient patients. So unfortunately, what is happening in the immunotherapy world is 85% of the colon cancer patients do not respond to immune checkpoint inhibitors because they have microsatellite stable type, which are predominantly they're having a proficient mismatch repair. So in one hand, like 15% of the colon cancer patients have a high potential to get cured of their disease, but then on the other hand, 85% of the patients, there's a chance that the immunotherapy could work, but at this point, they are not even candidates for immunotherapy. So there's something missing here in terms of, okay, how we can make predominantly the 85% of the colorectal cancer patients as a candidate for immunotherapy. That is the basis for my lab research, what we are trying to understand in our lab as a mechanism, how this can really happen and how we can bring these patients as a candidate. So I'll not go into the details of all the mechanisms and all those things. I'm going to give a broader picture view of what we are working in the lab as a research for colorectal cancer. So without going into the details of last 5 years of our work, all the graduate students in the lab and postdoctoral fellows in the lab, they worked tirelessly on this project. And what we really discovered was the colorectal cancer pretty much prevents the immune response by stopping the T cell function pretty much in the tumor environment. So what we call is like T cell co-stimulation, which is being like affected in the tumor environment. So how it is doing is This is, for example, this is a cancer tissue. As you can see, these are infiltrated with many of the immune cell types. Here, primarily shown in blue are like some of the T cells, which can kill the cancer cells, and some of the green cells here, which are like nothing but antigen presenting cells, such as dendritic cells. So these are all generally present in a tumor environment. Maybe the numbers are lower and higher depending on the immunogenicity of the tumor, but they are present in that environment. But what we discovered in the lab is these cancer cells, they secrete something called exosomes. These are, to simplistic term, they look like a garbage bag which are thrown out of the cells. And these small vesicles or bags, they contain the cellular contents, whatever the cancer cells has, they also package them in those small bags there. And what we found was these exosomes contain something called microRNAs. And these microRNAs can stop the function of the T cells, which can kill the cancer cells. So pretty much like a Trojan horse kind of system where they kind of hide in here, get into the cells and stop its function. So to kind of cut short a lot of functional data, what we found is the cancer cells secretes these exosomes with this microRNA 424. And when they are in the tumor environment, these immune cells, which can pretty much stop the growth of these cancers, they get affected because these microRNA target some of the key genes called CD28 and CD80, which are important co-stimulatory factors. And without that, there's a decreased co-stimulation and the immunotherapy will not work. So we have really nailed down the mechanism why cancer patients, at least at this point, like 85% of the colon cancer patients are not responding to immunotherapy in terms of the immune checkpoint inhibitors. So this work was published in Gastroenterology, one of the leading journals in the gastroenterology field. So, so mechanistically, we have understood why the cancer is kind of escaping the immune response. The next thing, what we also wanted to understand is, okay, of course, the vesicles or exosomes, they carry all this cancer cargoes. They, whatever the cancer cells has, they carry them. And like we discussed, they carry the immune suppressive factors. But what is also interesting is they carry tumor antigens, meaning any mutated proteins or peptides which can stimulate the immune response, these exosomes carry them. But along with that, they can also carry the immune suppressive factors. Because of the immune suppressive factors, these antigens which can stimulate the immune response are not really doing its job. So what we have been like kind of developing in the lab is how we can effectively make use of these exosomes to stimulate the immune response because they carry the tumor antigens in them. So one way to do that is to remove the immune suppressive factors which are present. And in our case, we are the microRNA 424, which regulates the T cell function, dendritic cell function. We can remove them and allow the tumor antigens to do its job. So we have done a lot of work engineering the tumor cells and deriving these exosomes and then testing in various animal models and to see whether this can be translated into in a clinical setup. So once we do that and once we create this immune response in the patients, then the immunotherapy can be added to these patients and then thereby the immunotherapy could work in these individuals. So towards that, we created a model system. So like any other treatment kind of testing, you need the right model to understand how the treatment could work more effectively and how the response can be measured. And colon cancer is kind of a little difficult model because it happens in colon. So you need to establish tumors in the colon so that we can treat those tumors in an orthotropic location, meaning actually in the colon, not below the skin or elsewhere in the body. So we are having this mouse models where we can develop advanced stage disease, and this disease also have metastatic potential. So you can see this metastasis happening in the liver. So in these models, which mimics a very late stage tumors, we can use this and see whether the immunotherapy are working or the modified exosomes are working. Again, you can see these are high-grade tumors, which also mimics the late stage. So using that model, we have tested our exosomes which we have removed the immune suppressive factors. And here we have tested with like only 2 doses of injection in the first week of the modified EVs or engineered EVs, and followed by the immunotherapy, the immune checkpoint inhibitors. And again, you can see these models where all these animals are having this advanced stage disease, and they are treated with different combinations of drugs. Here we are treating with controls, and here these animals are given only with the immune checkpoint inhibitors. And here the wild type EVs, meaning they are having the immune suppressive microRNAs and the immune checkpoint. And here are the control EVs, which also include the microRNAs. And here the EVs are devoid or lacking the microRNAs. So without the immune suppressive factor in combination with immunotherapy, like immune checkpoint inhibitors, you can see the disease burden is much lower compared to these other animals in the 3 weeks time period that we followed these animals, which translates to a lower amount of metastasis to liver in terms of the liver nodules. And also it translates to the better survival of these animals. Remember, we are starting with a very advanced stage disease, and the survival is better compared to the other groups here. So this has really given us a hope that combining the immune therapy, which is anti-PD-1, along with the engineered EVs or exosomes, you can improve the survival in these individuals with an advanced stage disease. And the next step is to see whether these animals have really an improved immune response. So we did extensive immune analysis. As you can see, there's a very enrichment of the T cells in the tumor environment. These T cells are very actively functioning, and they are making these enzymes that can really kill the cancer cells in that environment. So pretty much like taking these cold tumors and behave like a hot tumor. So these tumors, which are immunologically in cold, now after this treatment with combination, they become a hot tumor. So this has really provided us a hope that we can take this to the next level to the clinics, and many patients could be benefited. But to kind of do that, we need to have a kind of a patent filed, and then because this is a commercial process where the next level has to be like kind of an industry scale development of this approach, so that can be translated to clinics. So we have patented this approach in terms of moving to the clinics. So then now we can ask questions like, okay, how we can really kind of develop these engineered EVs. So the sources of this engineered EVs can be from the tumor cell lines. These cell lines are already established, and there is a library of cell lines available. And that means we can create off-the-shelf engineered EVs, or we can also develop this patient-derived tumor organoids. And you can see this looks like a cross section of a colon. So this is a pretty much a mini colon growing in a petri dish in the laboratory, in our laboratory. So we can isolate exosomes from the patient's derived organoids, and then that means this can be used as a personalized therapy. And here tumor heterogeneity may not be a problem. So we are developing these tools and methods to really move things from lab to kind of clinic. So bench to bedside is our kind of mantra that we are thinking here. So with all these things, colon cancer is like something which people come with a different kind of outcomes, mutation profile. So we are also developing a tool where what we call like Evolve TM, where we can do the artificial intelligence-based patient selections, basically looking at the mutation profiles of the patients and matching these patients to the library of EVs that we have, so that they will have a better response. And then we can combine the immune checkpoint blockade therapy for a better stimulation and sustenance of the T cell activation to kind of take care of the tumors. So this is what we are envisioning, how we can really provide the best treatment option that is available using this combination of engineered exosomes with immune checkpoint inhibitors, and then the patients may benefit. So this is all well and good, but there's also like more complexity to the colon cancer. As we all know, gut environment is enriched with a lot of bacteria, which we collectively call gut microbiome. And microbiome also plays a major role in controlling the tumor growth. Not only that, it is also controlling the immune response and immunotherapy response to these patients. So unless we understand what is really happening in terms of the interactions of the tumor cells and then the various immune cells and then the microbiota, what is called collectively all the microorganisms in the gut tract, it would be really challenging to really kind of make the patients to respond in a long term, the tumor response or treatment response in the long term. So in our lab, we're also trying to understand all the crosstalk between the tumor cells and the microbiota and how this microbiota can also control immune response. So what we have recently discovered in the lab so far is some of the microbiome, they secrete specifically like kind of increased essential amino acids. And these essential amino acids are very critical for some of the immune cells where not all immune cells are good. Some of the immune cells could be also be like kind of bad to stop the immune response. For example, macrophages, where specifically there are like two types of macrophages for general purposes, type 1 and type 2. If you have more of the type 2 macrophages, they can put a brake on the immune system. And what is happening in that environment is this bacterial secreted amino acids are essentially taken by the macrophages type 2, and they put a brake on the T cell functioning and then T cell proliferation. So thereby the tumor is effectively kind of continuing to grow. So these are the things that we are recently developing or trying to understand in the lab. And if you are interested, we can also share some of the recent review articles and another publications from our lab. So in a big picture, we have created various model systems like cell-based tumor models or tissue-based. We can take the colon cancer patients' tumor tissues, implant them in animals, and then trying to see how our treatment response, our treatment can be like kind of used in these models. We're also using orthotropic-based models where we can implant cells in the colon or colon wall directly and then see whether they are playing a role. We are also trying to humanize this mouse with microbiome, human microbiome, in terms of understanding how the microbiome is also regulating the immune response. So with all these different model systems in the lab, we are one step closer to developing therapies which could be more effective in the near future. And like I said, treatment response depends on the right combination of the treatments. And not only that, also the timing and finding the right patients. Only with this right combinations, we can achieve the better long-term treatment response. So all these factors which we are trying to understand in our laboratory, like for example, the immune cells in the tumor environment, and what are the metabolic fractions coming from the food that we take, and the microbiome that also contributes to these metabolites, and how these exosomes put the brakes on the immune response, and all these interactions we are trying to understand. So collectively, the things that we discovered in the lab will be moving towards the clinic so quickly and effectively. So with that, I'll stop and then I'll transfer to Mike so that he can also share his experience.
Thank you so much, Dr. Subramanian, for sharing your leading work in colon cancer research. So next, I would like to introduce you all our second speaker of the day, Mike Neeson. Mike lives in Stillwater with his wife, Patty, of 30 years. They have 2 adult children, Nick and Ben, both of whom are actually graduates of the University of Minnesota. Mike retired 4 years ago from Anderson Window and Door Corp after a 30-year career. Mike and his wife, Patty, like to travel, play golf, ski, tennis, and bike. He enjoys spending time volunteering with cancer patients as they deal with chemo, radiation, and surgery. Mike and Patty, with another couple, are involved in charity work to raise funds for colon cancer research at the University of Minnesota. To date, they have raised over $1 million to fund research at our very own institution. This is incredible. Mike, thank you so much for all of the work and the support that you do for cancer research. I am so honored to have you here today, and I would like to pass it over to you. Please tell us how your colon cancer journey began.
All right, thank you very much. My medical story really started back in 2008 in the spring. I was 45 years old. I was in incredibly good health. Didn't have any existing conditions or anything. And that spring, I started to have some digestive problems, maybe some kind of odd bowel problems, even kind of bloating, things like that. And that was obviously unusual for me. And it went on for, a couple months. And so I went into my local clinic, explained my symptoms, and it was determined I was probably lactose intolerant and had some, dairy issues. So I changed my diet for a couple months, and it didn't really get any better. So I went back in, explained my symptoms again to the same GP, and he gave it some thought, and he said, well, based on the symptoms you've said, maybe it's gluten. And so I changed my diet again, and that didn't really help either. Actually, the symptoms got progressively worse. By August, I was actually even starting to have blood in my stool. So he had said, you're 45 years old, you're probably going to be due for a colonoscopy. At that time, the screening age was 50. But he said, why don't we do one now? And so I said, sure, that makes sense. Let's see what's going on. And so I went in on September 18th, excuse me, and had my colonoscopy. And when I woke up out of the sedation, I remember him showing me a photo, and he said, this is your colon. And he said he had identified about a 1-inch mass in there. And he said they did a biopsy that, the results would be back in a few days. But he said, based on his experience, it was malignant. And so at that time, my wife was working in the medical field for a surgical instruments company. And that afternoon, she got on the phone, started to call colleagues and friends in the industry, and asked for recommendations. And the same name kept coming up, David Rothenberger here in the Twin Cities at the University of Minnesota. And so we were fortunate. My wife got us in on an appointment, and obviously he's extremely busy, but we got in within a week to see him. And by this time, the local clinic had confirmed through the biopsy that my tumor was malignant. But Dr. Rothenberger said, we'll schedule an MRI, a CT scan, and a PET scan. And so I got all three of those tests scheduled. And, I think I had them all completed within maybe 7 to 10 days. And so we went back to see Dr. Rothenberger. He had all the scans and imaging. And he confirmed not only did I have colon cancer, but it was advanced stage colon cancer. And he explained that really the protocol would be for 8 weeks of chemotherapy and then 8 weeks of radiation. And the 8 weeks of radiation was every day, Monday through Friday. So it was 40 radiation treatments along with the chemotherapy. And he also explained at that time, due to the location of my tumor, it was extremely low in my colon, almost really in my rectum, that I was going to have a colostomy bag, because once they took the tumor out and the surrounding tissue, there just wasn't going to be enough tissue or muscle left to hold in the stool. So this has really went from, bad to, in my opinion, right, even worse. And so I have to deal with the chemo, the radiation, and the thought, of a colostomy bag. And so I get through, the treatments there. And I had asked, Dr. Chen was my radiologist at the U, and then Dr. Greeno was my oncologist at the U. And I remember asking him early on, probably what most of you would ask too. I said, what are my odds, or what are my chances? And he said something to me that stuck with me, forever here in the last, 15 years. It was, profound, and I've always remembered it. And I said, hey, what are my odds? What are my chances? And he said, Mike, unless I can find a whole pool of 45-year-old non-smokers, non-diabetic, healthy people, he goes, I really don't know what your odds or chances are. But he said, at the end of the day, Mike, he said, your odds are 0 or they're 100. You can't be 20% alive. And that just always really stuck with me. So again, I got through all of October, all November. That was the 8 weeks of my treatments. In early December, I went in for an MRI again. Finally, some good news. The tumor had shrunk pretty significantly through all those treatments. Again, that was early December. And so now it's late December, and my surgery was scheduled for January 6th. it was going to be a major surgery to remove the tumor, to have the colostomy. And so I went in late in December, and I had to go in and get what's called marked for the colostomy bag, because they don't want it where your belt is or where your pants and are going to interfere, things like that. It's your lower abdomen. So I went in and they marked me on both my left and my right side, which I thought was odd. But then I found out that due to the 40 radiation treatments, I actually had collateral damage in my bladder. And so they were going to have to remove my bladder. So then I met my urology surgeon, Dr. Sean Elliott at the U. So now I'm going to have, 2 bags. And so I go in for the surgery on the 6th of January. It's a 10.5-hour surgery. Short for me, right? Pretty long day for my family. And I spend the next 21 days in the hospital at the U. And, due to the significance of the surgery, and I get home, and now I kind of have to, learn on my own, right? My new body and how it's changed and functions and things like that. And so maybe I'm home a month, and now I go back and I meet with Dr. Greeno, my oncologist. He wants to know how things are going. I said, I'm adapting and things and adjusting. And he said, it's good. We're going to start another 26 weeks of cleanup chemo. And to remind you, 26 weeks, right? That's half a year. And so, I go back, start the chemo, and it was the same chemo I was on in the fall. And so, I get into that, for a couple months, and I start going back to work. And about the end of April, I'm going back, 2 hours a day. I'm slowly increasing it to, 4 hours, and then, working my way up. And, by summer of 2009, things are getting back to normal a little bit. I'm working more, not full time yet, but working more. I'm back to the golf course because that's, one of my passions. And I'm starting to see friends at the golf course who I hadn't seen for now. It's almost a year, because I really stopped playing back in, September. And so now it's July. And, some of my friends at the golf course are like, how are you doing and stuff? And I'm like, I'm doing pretty well. And they're like, how's your golf game? And I said, actually, it's really good. I said, I haven't hit a bad shot yet. And I'd get kind of an eyebrow raise, and they're like, you haven't hit a bad shot yet? I'm like, no. And what they didn't understand is, for me, it could go in the pond, it could go in the bunker, it could go out of bounds, right? At this point, I'm just happy to be playing. There's really no such thing as bad shots anymore. So that October, I go back to work full time, but it's been an entire year. And so, the next 2 years from there are pretty normal. I'm going in to see Dr. Greeno every quarter, every 12 weeks for blood work, CT scan, imaging, things like that. And the next 2 years, like I said, are pretty uneventful. I'm back at life and things like that. But then in the fall of 2011, I get a cough, and I just can't shake it. And at this point, I've actually migrated to every 6-month checkups, because I'm done with 2 years. But I can't shake this cough. And so I go in, for my checkup early, and sure enough, they do a CT scan and PET scan, and the colon cancer has metastasized to my lung. And so I meet my third surgeon now, Dr. Maddox, and he explains they're going to do a partial lobectomy and remove part of my right lung. And actually, I went in on a Friday morning and had the procedure. And by Saturday night, I was discharged. So it was literally like 36 hours. And so then I go back and I see my friend about 2 weeks later, Dr. Greeno, and he says, how'd it go? And I said, actually, it was great compared to a 21-day hospital stay earlier. I said, this 36 hours, I was kind of in and out. And he said, well, I'm glad to hear things went well. He said, because we're going to start another 26 weeks, another half a year chemo. Except this time it's going to be 2 types of chemo, Xeloda, that I had been on the 2 earlier times, and then another one called oxaliplatin. And so now I'm on double chemo for 6 months. And so that starts in about March of 2012. But before I can even get through that 6 months, in about May, I start having some real serious kidney pain. And so I go in and I see Dr. Elliott, my urology surgeon, and he does some scans, imaging, and he finds that my ureters, which are like two straws that drain your kidneys and the urine goes through, they have what they call strictures or scar tissue from all the radiation. So the urine really can't get out of my kidneys very well and through these straws. So he explains we're going to put stents in these ureters to keep the ureter walls open so the urine can go through. And I remember, I'm not a medical professional by any means. And I remember, him telling me this, and he said, do you understand? And I remember taking my fingers like this to about an inch. And I said, yeah, I get the concept of a stent. I have a friend or two, with heart stents. And so again, I'm like, yeah, I get the concept. And he said, I don't think you do. He said, these are 16 inches long. And he said, we're going to replace 2 of them every 60 days for the rest of your life. And I'm like, yep, you're right. I didn't quite have that concept. And so, it's been 11 years now since I've had the stents. I go in every 60 days. I was in last Thursday, had them replaced. So roughly in 11 years, I've probably had them done 80 times at the U. So I kind of kid that I'm, kind of a platinum customer over there. But I'm happy to say, other than that, the 11 years since then, I'm cancer-free. I enjoy my life. A sincere thank you to Dr. Chen, Dr. Greeno, Dr. Maddox, Sean Elliott, Dr. Elliott, Dr. Rothenberger, and my wife. So thank you.
Mike, I don't even know what to say. Thank you so much for sharing your incredible journey and story with us today. I am moved, and so I am just honored that you were able to share with everyone here today. So thank you so much.
Thank you.
Thanks, Mike. I think you are the hope. You are the hope that everyone wants to see. Right? I think at the end, everything is going to be fine, and the advancements in terms of research, everyone is working towards that, and you are the hope for everything. You are the hope.
And you are 11 years cancer-free.
Yeah.
Amazing. So we will transition to our Q&A and our discussion for the day. And Mike, in the comments, you are getting some kudos. So someone just said, thank you, Mike, for sharing your story. Amazing and incredible. So with, I just need a minute here, but so Dr. Subramanian, you mentioned a fire. So we always get a fire started before we start Q&A for the fireside chat. So let me share that. Because it is not a fireside chat without starting a fire and a nice warm glass of your beverage of choice. But we'll go ahead and start with some of these questions. So we've had quite a few questions that have been submitted before the Zoom today. Mm-hmm. And actually, I just see a question right now that says, just asks about just the screening age and recommendation. So how can Mike's story encourage the new 45-year-old screening guidelines with the public?
So first of all, I'm not a clinician or a physician to directly answer direct questions in terms of the things, but what my understanding in some of those colon cancer screening. So it was 50 years before the American Cancer Society really changed it to 45 years. And now our understanding is there's going to be an increase of 140%, mark this number, 140% increase in early onset of colorectal cancer. That's going to be like 27,000 individuals who will be diagnosed with colon cancer every year who are less than 45 years of age. So this really tells us the screening and then the diagnosis has to be one of the major tools that has to be really brought in everyone's mind, that early diagnosis is important. And as we know all the time, right, prevention is better than cure. Mm-hmm. So how we can really kind of think through this process, any symptoms, any blood in the stool samples, small things, any weight, uncontrolled weight loss or anything that one can think through, right? These are something that we cannot take it lightly. We have to immediately seek attention of the clinician doctors and make sure that, okay, you explain them what's going on and let them make the choice for whatever is best for you in terms of whether we want to go for the colonoscopy or any stool-based diagnosis or screening, those kind of things. So that's the first step, to reach out to the physicians and then explain what's going on. So don't take things anything lighter. So I think this is the first step to kind of think through this.
And also, just on that note, so when we say that colon cancer can be prevented, what do we mean by that? So what happens when why do people do colonoscopies, and what's the purpose of them?
So if you look at colon cancer development in general, it starts like something called polyps. These are small benign tumor growths in the colon. Doesn't matter where it is. It could be in the right side, in the transverse, or left side, in the sigmoid region, or the rectal regions, right? So anywhere in the large intestine, this can be a small tumor growth, which are benign tumors. And most of the time, it takes like up to like decade for the benign tumors to become malignant. So that is, at least it's giving that much time for us to do something, right? So during this time, it starts as a benign tumor, but if you're not careful enough, you're not doing any screening, it has the potential to become a malignant tumor, which has the potential for metastasis, right? So that's where things can go really quickly wrong. So the benign tumors provides us enough time for doing a screening. So I think this is why the screening becomes so important in terms of colonoscopy. Like I said, these polyps can come anywhere in large intestine. So doing a colonoscopy will allow us to doctors to see this, whether there's any benign growth in the colon, and then it's a very simple procedure to clip the tumor out. While they're doing the endoscopy, there are instruments where they can just remove the benign tumor just like that, right? So that you are removing this benign tumor, and then no longer that's a threat for into developing a full-fledged cancer. And then the doctor determines, okay, depending on what they see during the endoscopy, they can say, okay, you come to the clinic every 5 years or 10 years, depending on what they kind of measure in terms of the tumor and the numbers of polyps they identify, and also the age factors. So they consider a number of things to kind of get back to you to the clinics for routine screening. So it will be helpful. The other thing is, most of the colon cancer are sporadic in nature, meaning like they happen spontaneously. And many times is also genetics based on our parents, right, who has some kind of mutation. We generally call Lynch syndrome. If there is any specific mutation that could be inherited, that puts under a very high risk category. So in those conditions, I think routine screening and then all those things will be really important here. So, but screening is everything here.
And I think what we can take away from even just Mike's story is that at the time you weren't even, Mike, at the recommended age for screening, but because you were in tune with your body, you knew what was abnormal for you, and you went and sought care. You talked to your physician about it, who then recommended that you do the screening. So I think what we can take from your story, Mike, is even if you are not at the recommended age of screening for colon cancer, talk to your provider if you see and detect any of the symptoms that Dr. Subramanian had mentioned. And then with that, also with the screening guide being lowered from 40 or 50 to 45, Dr. Subramanian, can you talk a little about why that is? Is it because we started seeing an onset of colorectal cancer cases in younger patients?
Yeah, that's true. So like I mentioned before, there's a 140% potential increase in early onset of colon cancer that pretty much translates to 27,000 individual patients, right? So with that number, and again, the American Cancer Society has brought the screening age from 50 to 45. So that's one of the reasons why the screening age was reduced now, right? But again, with early onset, of course, there's a lot of things they're doing in terms of risk and benefits. So whether the number of screening they do will really translate into a kind of things in the early stage, like younger individuals, right? So that's one of the reasons why people with like 30 years or 24 years or like anything above like 30, they are not recommended for screening at this point. Mm-hmm. So, but like we discussed, having any symptoms, any blood in the stool sample or any discomfort, any weight loss, that's a trigger, right? So we never know who the cancer, who is going to get a cancer at early stage, right? So even though there's no screening recommended at early ages like 30 and 40, but it's better to really follow our body and then to really take this as an important kind of indicators and then talk with your doctor. So.
Thank you. And Mike, you actually had a question in the chat. So when you say, are you cancer free or in remission?
I would guess I'm cancer free after 11 years. Like I said, I haven't had any symptoms. I actually haven't went in for any scans or imaging for many, many years. But as I mentioned, I'm over there every 60 days for my stent procedure. And as you said, I'm pretty in tune with my body. So as Subree said, if I lost weight, if there were different things, I think I'd have, some indication after the different situations I've been through. But again, screening, is imperative. And as Subree said, when they did the pathology on my tumor, it was 8 to 10 years old. And so, right until the end, I really didn't have any symptoms for, maybe, almost the entire time until the end.
Did you have a family history? I know sometimes that could increase your risk for developing.
No, I don't, not that I'm aware of. And when Subree talks about polyps and things, obviously, that's important. I've had one polyp my whole life, and obviously I go in for colonoscopies, pretty often, since I was originally diagnosed, and I've only had the one. So Dr. Rothenberg one time said, "You've had one," and it kind of went nuclear. So, again, screening is everything, right?
I forget that there's a dog. But so Mike, tell me, how did your diagnosis change you? Has it changed how you look at yourself, your outlook on life?
Oh yeah, I don't think anybody could go through that journey without being impacted. I've always been a real positive person anyway. But I think, when you go through something like that and you kind of look at your mortality, little things, I'll say, right? We all have appliances that break down, we have car issues, things like that. Those used to be maybe a bigger deal, and now they're just not, right? So you prioritize, and I guess you really put things in perspective on what's important and what's just not.
Were you going to say something, Dr. Subramanian?
Yeah, I think it's true. Whatever we are hearing from Mike, right? It's a lesson for everyone to kind of go for screening.
And I mean, on that note, so Mike, now with your diagnosis and kind of your journey, how have you been able to talk to your loved ones about colon cancer and why it's so important to get screened?
Actually, as you mentioned, we have two sons, 24 and 26. Our 26-year-old just had his first colonoscopy back here in December because, as I mentioned, I was 45. And so Dr. Rothenberger had determined, again, through the pathology that maybe mine started right at 35 roughly. So then they back it up 10 years from there. So really when, our oldest son, who, at 25 here, he just turned 26 in January, they really recommended at 25. Now he went through and he was fine. And he'll go back, in 5 years. And when I got sick, originally diagnosed 15 years ago in 2008, some of the genetic testing wasn't available that is now. And Dr. Madoff from the U, who's a recently retired colorectal surgeon, got me in about 5 years ago for some genetic testing, again, that wasn't available back early on. And, it doesn't look like from the genetic testing they did that in my case, it's hereditary. I don't, according to the information I got back, our two sons are no more likely than anybody else, but that doesn't mean, they won't be prudent, right, and go in for screenings, because as Subree said, it's really addressable early, right? You go in for a colonoscopy, which people think might be a big deal. It's just not, right? And almost every time, if you have a polyp, they don't tell you to come back, right? They just take it off right then and there. So, you know, at times I'm kind of the Katie Couric of get your colonoscopy.
Love that. It is the best prevention tool that we have for colon cancer. So thank you for sharing that. Also, I mean, stressing the importance of family history, knowing your family history, and making sure that your loved ones and families are getting screened if they are able to. And so we'll pivot just a little bit. We had some questions for you, Dr. Subramanian, that were submitted about kind of addressing some of the inequities in colon cancer screening and mortality. So if you can share those with us.
Yeah, inequities in cancer is a kind of main issue at this point, right? Because specifically like socioeconomic status and then like access to the healthcare, a lot of things have an impact. So even if you have like facilities for screening, but accessibility, not having that real time and then the kind of support, right? The system can also have some kind of impact on the inequities. So there's so many factors that can really contribute to cancer development, and the food that we eat, socioeconomic status, like I mentioned before, high fat, low fiber food, fast food that we generally kind of we don't have time, and we don't have money to buy good quality food and all those things. Everything really contributes. So keeping our gut microbiome happy, right? Will really provide us and return back a lot of favors for us. So how do we keep our gut microbiome happy? By eating good food, high fiber rich food, keeping the fat and then the kind of red meat levels lower. So that will really improving some kind of screening and outcomes and the disparity. It can reduce the cancer disparity. This is a very challenging topic. If we know what exactly contributes to cancer disparity, and especially colon cancer disparity, that's easy to target, but because it's so complex, multiple factors are involved, and there could be like a genetic background, there could be a lot of other things contributing to it.
And with that, we know that African Americans have some of the highest rates of colorectal cancer. Do you know if there's any research right now looking into why that is?
Yes, so this research is happening at every level in our institute, at the national level, at NIH. NIH is also like really putting more emphasis on looking specifically on why there is an increase of colorectal cancer in African Americans, and our institute, we are also trying to reach out in terms of the patient population, in like understanding like why specific fraction of patients get this increased incidence of colon cancer. So like we discussed, it could be specifically genetics or it could be other factors. So that's what we are trying to figure it out. What is really contributing to that disparity and how we can narrow it down and how we can target that.
Great, thank you. And so I know you have both talked about or mentioned this word metastasis. Could you explain to us what that means? And if is it likely for colon cancer to spread to other parts of the body?
Yeah, in a simplistic term, cancer metastasis, and in terms of understanding the biology, is very complicated. But in a simplistic term, from a primary location where the tumor initially starts, when it moves to a distant location, in terms of, for example, in colon cancer, it primarily to the liver or lungs. Sometimes it could be out to the brain. So it has different regions in the body that it can also go to as a secondary location, right? So when it moves from the primary location to the secondary location, that's what we call it as a metastasis. So, but that's also like kind of, it gives some time for the primary tumor to kind of go into the process of metastasis, right? So it doesn't happen overnight, but it's a lot of things happens. It's primarily puts a brake on the immune system. It kind of goes to the new location. It's a seed and the soil kind of concept where this tumor prepares the land and then established in a new location. And then it hides from the immune system for some times, and then it really shows up as a secondary tumors. So that's why when doctors or surgeons, when they remove the primary tumor, they also want to make sure there's no any like local metastasis or lymph node metastasis. They also remove some of the regional lymph nodes associated with the primary tumor so that it doesn't happen. But one issue with metastasis is because it spreads to other organs, there could be like more lung nodules or more liver nodules. And unfortunately, if there are more nodules that can be really surgically removed, then metastasis surgically cannot be removed, right? So that's a challenge there with metastasis.
Thank you. So, Mike, this question is actually for you. So, what resources have you found most helpful during your most difficult times?
I think colon cancer is different. When people sometimes have diabetes and things like that, you typically know somebody maybe that you work with, or for colon cancer it was a little different. I didn't know anyone, so it wasn't as easy just to go talk to somebody. And I was cautioned by Dr. Rothenberger early on, to kind of stay off the internet, and so I did. I'm not a big internet guy as much as I am reader, so I guess as far as resources, I probably did more reading to educate myself. But a lot of the reading I did, and it's not right or wrong, because I work with a lot of ostomy patients, colon cancer patients too, just kind of from a support purpose, not obviously from a medical purpose. But I trusted the medical team implicitly. And I wanted to read, kind of from my own, not to propose more solutions or understand the research a lot. Some of the reading I did was just more how to deal with it mentally, because there's a physical aspect to it that in some ways is almost easier to deal with than the mental. So the reading helped me, with some of the mental aspects of just trying to take it, really a day at a time, because for me, it was an extremely long, 3 years, that first 3 years. And what I try to tell some of the people I work with, there's a lot of up and downs, and you have to deal with that. It's you get good scans or imaging, and you're tempted to kind of, what I'll say, want to spike the football and celebrate. And that's okay within reason. But, even for survivors, it's not linear, where it's, just kind of, there's up and downs along that, linear path. And like I said, you get good scans, you should be happy. And when you get bad scans or blood work, sometimes, again, based on my experience, you don't want to get too despondent, because I would go in every quarter for a long time, for imaging. And sometimes they'd see something on a CT scan and they weren't sure what it was. And when I went in 12 weeks later, it was gone. Could be bacteria, could be different things. So I think my advice, and some of it, is, you don't get too high, don't get too low. Take it a day at a time. It's a long journey. And that worked best for me.
Awesome. And you have so many praises and comments that were just submitted. So thank you so much, Mike. And you have such an amazing support system too. Just what you've shared about some of the work that you and your wife Patty do, and how you give back. So thank you so much. So we're out of time, but I do want to ask just one more question for both of you. So what is the one takeaway message from our talk today and our conversation today that you would like our audience to know? And so we'll just quickly do this so we don't keep folks for any longer here, to just be.
Go ahead, Subree.
So again, if it's a one takeaway message.
One takeaway message from your research, from just our conversation today, that you want our audience to know.
It's prevention is better than cure. Screening as soon as possible when you are in the right age. Don't delay the screening process. And that will take little time, and then it will save a lot of things, especially mentally. You can be free, not nearly worrying about for next 5-10 years about the process, right? So that's really helpful. And also to understand, if I may have a chance, so much of research and development has been happened in the last decade or so. And the immunotherapy, the understanding of our microbiome, the understanding of the metabolic contributions and all those things has really come a long way. So I would imagine that cure for colon cancer, for that matter, for any cancer, is not too long from now. So that's a hope for everyone. And I think it's so wonderful to have that.
And it's all of the amazing work that you're doing, and just all of our researchers at the Masonic Cancer Center. So thank you, Mike.
Yeah, I would say the same thing. You know, even if you feel great, no family history, at 45, go in. And anybody who has trepidation around getting a colonoscopy, right, it doesn't sound appealing. On a scale of 1 to 10, it's a 1. It's just nothing. So go get it done.
Love that. Thank you. So we're out of time. We're actually a little bit over time. So just want to be respectful of everyone's time and sorry for keeping you a little bit longer. But Dr. Subramanian and Mike, thank you so much for being here with us to help us spread awareness about colon cancer this month and just for all that you do. So you will all receive an email shortly following today's program with a link to complete a survey. We would appreciate it if you could just spend a few minutes to provide us with your thoughts and feedback because they really do help us improve and plan future fireside chats. And after completing the survey, you can enter to win a prize pack filled with goodies from the Masonic Cancer Center. And so everyone who tuned in today will be entered into a drawing to win a $25 gift card. So keep your eye out. Keep your eye on your email for that because 2 lucky winners will receive a $25 gift card. And join us next month for our fireside chat on April 18th from 3 to 4 in recognition of National Minority Cancer Awareness Month to learn about the current state of cancer disparities in Minnesota and hear about some of the exciting and impactful research being conducted by minority population communities across our state in collaboration with researchers at the Masonic Cancer Center at the University of Minnesota. And be sure to follow us on our social media platforms to stay up to date on all the events that the Office of Community Outreach and Engagement hosts. Thank you everyone for joining us today and we hope to see you soon. Thanks again.
Thanks,
Bye.
Amit. I think
Thank
it's
you.
wonderful that you took the time to organize this fireside chat. Thank you.
Thank you so much for being here. Bye now.